हेलो फ्रेंड्स, हम डिस्कस करेंगे जेनेटिक बेस ऑफ इनहेरिटेंस के नोट्स जो कि काफ़ी इम्पोर्टेंट है पेपर के लिए। सो फर्स्ट ऑफ ऑल वी विल डिस्कस सिलेबस। इसमें आता है इंट्रोडक्शन में मेंडेलियन इनहेरिटेंस, मेंडेलियज्म टर्मिनोलॉजी, मोनोहाइब्रिड क्रॉस एंड रेशो, डाइहाइब्रिड क्रॉस एंड रेशो, मेंडल्स लॉ ऑफ इनहेरिटेंस, law of dominance, law of segregation, law of independent assortment, test cross and back cross, deviation from Mendelian ratios. Introduction देखेंगे. For a long time, the science of genetics was nothing but speculation. Initially, it had been accepted that inheritance of characters takes place by blending inheritance. Blood or heredity units of both parents get mixed in offspring. Mendel was the first to think of hereditary factors as particles that would coexist but not blend. The first scientific explanation regarding the mode of transmission of characters was given by Mendel in 1866. So therefore, Mendel therefore rightly called father of genetics, and, and his work on the transmission of characters from generation to generation is called Mendelism. Some plants and animals show different inheritance patterns for few characters in which Mendelian ratio deviate described as post-Mendelian genetics. तुम क्वेश्चंस और आंसर देखेंगे जो भी इस चैप्टर में है. सो पहला क्वेश्चन आता है इसमें, प्रैक्टि राइट नोट ऑन बायोग्राफी ऑफ ग्रेगर जॉन मेंडल 1822 टू 1884 में 84 तक इनका पीरियड है. So, first point is Mendel was born on 22 July 1822 in Silesian villager near Hessendorf of Austria, which is now a part of Czechoslovakia, in the family of poor farmer. Second point is Mendel completed graduation and joined the Augustinian monastery of Brunn as a priest. Third point is Mendel was sent to University of Vienna for higher studies in physics, mathematics and natural science. After completion of higher studies, he returned and again started to work as a priest and teacher. Fourth point is Mendel started his famous experiment of hybridization on garden pea in 1856. Fifth point, in 1865, Mendel presented his work in a paper, Experiment in Plant Hybridization. Experiment in Plant Hybridization at the meeting of Natural History Society of Brunn, which later on publication in journal Annual Proceedings of Natural History Society in 1866, German language. Mendel's work remained unnoticed and he died in 1884. In 1900, Mendel's work was rediscovered independently by Hugo de Vries of Holland, Karl Koren of Germany, and Erich Schermack of Austria. De Vries published Mendel's research paper in journal Flora in 1901. Ninth point is Koren proposed three laws known as Mendel's law of inheritance, which was based on Mendel's findings. Next question is define following terms. पहला है जीन्स, दूसरा है जेनेटिक्स. तो जीन्स का है, जीन्स की डेफिनेशन, अ पर्टिकुलर सेगमेंट ऑफ डीएनए governing the unit character of heredity is called gene, and the genetics is the branch of biology which deals with the study of the heredity and variation is called genetics. The term genetic is coined by William Batson in 1906. Question third, define heredity and variation. Heredity, first point, the transmission of characters from one generation to the next or from one from parents to offspring is called heredity. Variation. The difference observed between the parents and offspring among offspring of the same parents and among the individual of same species is called variation. Fourth question is write a short note on like begets like. Like begets like. The tendency of organism to produce the individual of their own kind is called like begets like. Point first is it is a fundamental, long known principle of biology. Second, it is essential for the continuity of races. Third, the individual developed through a sexual reproduction show close similarities than sexually reproduced individuals. Examples say cat will produce kitten, dog will produce pups, pea plant seed will germinate and produce pea plant. Mango plant seed will germinate into mango plant. Question fifth is topic A Mendelism. Enlist 7 straight in P selected by Mendel. Answer. तो पहला था कैरेक्टर्स थे डोमिनेंट ट्रेट थे रिसेसिव ट्रेट थे तो पहला तो पॉइंट था स्टेम हाइट टॉल थी और डॉर्फ थे डोमिनेंट कैपिटल टी थी और स्मॉल टी ड्वार्फ थे यानी रिसेसिव सेकंड सीड कलर था येलो जो कि डोमिनेंट येलो था और रिसेसिव ट्रेट ग्रीन था और एक कॉटिलिडन था तीसरा पॉइंट आता है सीड शेप डोमिनेंट ट्रेट राउंड था और रिसेसिव ट्रेट रिंकल था फोर्थ है पॉड कलर डोमिनेंट ग्रीन रिसेसिव ट्रेट येलो पॉड शेप डोमिनेंट इन्फ्लेटेड एंड रिसेसिव कंस्ट्रिक्टेड सिक्स्थ है फ्लावर पोजीशन डोमिनेंट स्ट्रेट पे था एक्सियल और रिसेसिव स्ट्रेट पे था टर्मिनल विच इज डिनोटेड बाय स्मॉल ए फ्लावर कलर था कलर्ड which is capital C and recessive था white which is small c and seed coat color था dominant state colored and recessive state white. Sixth question is why did Mendel select P for his experiments? Explain the characteristics of P. Mendel's choice of P. Scientific name Pisum sativum plant. So Mendel's choice of pea plant for the study of heredity was perfect because pea plant shows following characters. First, the short life cycle. तो इसका पहला कारण था short life cycle. The pea plant is annual plant with short life cycle about 3 to 4 months. 3 generation per year. यानी 3 जनरेशन इसकी एक साल में हो सकती है 3 बार उगाया जा सकता है इसे एक साल में सो मेनी generations can be studied in short time. Second point था pollination. Pea plant is naturally self-pollinating as it has bisexual flowers. Flower size is large enough for easy emasculation required for artificial cross. तीसरा कैरेक्टर था contrasting character. The plant showed many contrasting character which are easy to distinguish. There are no intermediate character. Fourth, the seeds. It produces large number of seeds. Fifth is it is easy to handle and cultivate. Sixth, it produces fertile offsprings. 7.8 it requires small space for growth which enable Mendel to grow large number of plants in small plot of 70 into 7 meter behind church. अब कैरेक्टरिस्टिक्स ऑफ पी की देखेंगे पी फ्लावर हैव papilionaceous corolla that is butterfly like corolla. Second point is the corolla has 5 petals of which the innermost 2 petals are partially fused with each other to form a boat-shaped structure called keel or carina. The carina encloses the stamens and carpels. Third, stamens and carpels mature at the same time, as a result of which there is self-pollination. Fourth, stamens are 10 in number. The filament of 9 stamens are fused with each other while the 10th one is free. Diadelphous stamens, that is 9 plus 1. This makes the emasculation easier. For emasculation, the carina is open and the stamens are removed easily. Question 7 is enlist the classical method of symbolization. So the first filial generation is denoted by F1, second filial generation F2, third filial generation F3. डोमिनेंट को डिनोट किया जाता है कैपिटल टी से फॉर टॉल, कैपिटल वाई फॉर येलो। रिसेसिव को दर्शाता जाता है स्मॉल टी फॉर ड्वार्फ के लिए, ओनली स्मॉल टी, स्मॉल वाई फॉर ग्रीन। और मेल जो कैरेक्टर दर्शाया जाता है वो इस साइन से, और फीमेल दिस। Question 8 is describe the steps or procedure of Mendel's experiment with suitable example. The step or procedure of Mendel's experiment are as follows. First, ए में दिया है सिलेक्शन ऑफ मटेरियल. Mendel selected garden pea, that is Pisum sativum, for his experiments. The reason for selection were as follows. First point is garden pea, being an annual plant, completes its life cycle within 3 to 4 months. Owing to short lifespan, Mendel could get 3 to 4 generations in a year. Second point is many varieties of garden pea were available with contrasting characters such as tall versus dwarf plants, yellow versus green seeds, violet versus white flowers. Third point is flowers were large enough to facilitate easy emasculation for artificial cross and produce fertile offspring. बी में दिया है selection of characters. Mendel selected 7 pairs of contrasting characters for his experiment, which were as follows, जैसे दिखाई दे रहे हैं. Small t dominant recessive character, stem height, stem color, seed shape, pod shape, flower position, flower color, seed coat color. ये सभी डोमिनेंट और रिसेसिव दिखाए गए हैं. सी में आता है प्रोसीजर ऑफ द एक्सपेरिमेंट. So Mendel was very methodical in his experiment. He studied the inheritance of one trait at a time, unlike others who studied the organism as a whole. Then the studied the studied two characters and three characters respectively at a time, thus performing monohybrid, dihybrid, and trihybrid crosses. Second point is Mendel started his experiment with true breeding plants and maintained complete record of actual number of each type of offsprings for 3 generations. Mendel conducted many crosses to eliminate chance factor. Third point is Mendel conducted his experiment in the following 3 steps. First step is selection of parents and obtaining pure lines. Second point is artificial cross and the selected parents. Third point is selfing of F1 hybrid to raise F2 generations. Step 1 में था selection of parents and obtaining pure lines. Mendel selected pure lines that were locally available and ensured that the selected male and female parent plot were true breeding for the selected traits. by selfing them for 3 generations. Step to the artificial cross of the selected parents. Mendel emasculated means removal the stamens. Mendel emasculated the flower of the plant which he selected as female parent. After this, the pollen grain from the flower of the selected male parent were deposited on the stigma of the emasculated flower, that is artificial cross. Mendel crossed many flowers in this way, collected their seed, and raised hybrid representing F1 or first filial generation. Filial means offspring. Step 3 is selfing F1 hybrid to raise F2 generation. Mendel allowed self-pollination in F1 hybrid, collected seed from them and raised F2 or second filial generation. F3 or third filial generation were also raised by selfing F2 generation. कुछ टर्म्स जानेंगे जो इसमें दी गई है कैरेक्टर्स इट इज़ अ फीचर ऑफ द ऑर्गेनिज्म हाइट ऑफ स्टेम एग्जाम्पल में दूसरा है ट्रेट ट्रेट क्या होती है एन इनहेरिटेड कैरेक्टर एंड इट्स डिटेक्टेबल वेरिएंट उदाहरण में दी गई है टॉल और ड्वार्फ सम ट्रेट कैन बी इन्वायरमेंटली डिटरमाइंड और मॉडिफाइड तीसरी टर्म है जीन इट इज़ अ पर्टिकुलर सेगमेंट ऑफ डीएनए विच इज़ रिस्पॉन्सिबल फॉर द inheritance and expression of that character. The term was coined by Johnson. Dominant term dominant. It is an allele that expresses its trait even in the presence of an alternative allele. In heterozygous condition, only dominants express in hybrid. उदाहरण में एग्जाम्पल में दिया है टॉलनेस राउंड सीड इन पी प्लांट्स पांचवी टर्म है रिसेसिव इट इज एन एलिल विच इज नॉट एक्सप्रेस इन द प्रेशर सॉरी in the presence of an alternative allele that is in heterozygous condition it expresses only in the presence of another identical allele जैसे दिया गया है ड्वार्फनेस ग्रीन सीड कलर इन पी प्लांट सिक्स्थ है होमोजाइगस टर्म सिक्स टर्म होमोजाइगस an individual possessing similar alleles for a particular trait is called homozygous or pure for that trait. Homozygous breed true to the trait and produce only one type of gamete.